Bacterial cells are surrounded by a protective layer called the cell wall, which provides shape, rigidity, and protection from environmental. The cell wall is made up of a complex structure of molecules, including peptidoglycan, lipopolysaccharides, and teichoic acid. In this lecture, we will discuss the structure, function, and biosynthesis of bacterial cell wall. Structure of bacterial cell wall. Bacterial cell walls are composed of a complex network of macromolecules that provide structural support and protection. The main component of bacterial cell walls is a polymer called peptidoglycan, which consists of alternating units of N-acetylglucosamine, NAG, and N-acetylmuramic acid, NAM, connected. The peptide bonds are cross-linked by amino acids, forming a three-dimensional lattice that gives the cell wall its rigidity. In addition to peptidoglycan, some bacteria have an outer membrane composed of lipopolysaccharides, LPs, which are composed of lipid molecules and complex carbohydrates. LPs can be toxic to host cells and can cause an immune reaction. Other bacterial cell wall components include teichoic acids, which are acidic polymers that are covalently attached to peptidoglycan or the plasma membrane. Teichoic acids help to maintain the structural integrity of the cell wall and also play a role in cell adhesion. Function of bacterial cell wall. The primary function of bacterial cell walls is to provide support and protection to the bacterial cell. The cell wall helps to maintain the shape and rigidity of the cell, and also provides a barrier against environmental stresses such as osmotic pressure and chemicals. In addition, the bacterial cell wall plays an important role. Something went wrong. Interactions with other cells and the environment. The cell wall can contain surface proteins, lipids, and polysaccharides that allow bacteria to interact with host cells, other bacteria, and their. Bacterial cell walls are also important targets for antibiotics, as they are essential for bacterial survival and. Many antibiotics target the biosynthesis or function of the bacterial cell wall, making it an important area of research for the development of new biosynthesis of bacterial cell wall. The biosynthesis of bacterial cell walls is a complex process that involves the coordinated activity of multiple enzymes and The process begins with the synthesis of peptidoglycan precursors in the cytoplasm, which are then transported across the plasma membrane and into the periplasmic space. In the periplasm, the peptidoglycan precursors are assembled into long chains by glycosyltransferases and transpeptidases, which form the peptide crosslinks between adjacent. The assembly of peptidoglycan is a highly regulated process, and mutations in key genes can lead to defects in cell wall synthesis and cell. Many antibiotics target the biosynthesis of peptidoglycan, either by inhibiting the activity of key enzymes or by interfering with the transport of peptidoglycan precursors across. Conclusion. In summary, the bacterial cell wall is a complex structure that provides structural support, protection, and environmental sensing for bacterial cells. The primary component of the cell wall is peptidoglycan, which is assembled in the periplasmic space by a series of enzymes and proteins. The cell wall is an essential target for antibiotics, making it an important area of research for the development of new drugs to combat bacterial infections.